Now let's talk about blood groups and blood transfusion. So the very first time that the antigens, the ABO blood antigen gene was identified was in 1910, and really explains the incompatibilities in blood transfusions. The blood types are distinguished by the antigens that are found on the surface of the red blood cells. So what is an antigen and what is an antibody? An antigen is a molecule that can produce or generate an immune response. So gen means to generate something, anti means that immune response that the body will produce in response to this foreign body, what it can consider a foreign body. If the antigen, our immune system has the ability to identify our own antigens. These are known as self-antigen. So our immune system does not attack our own antigens, but it should be able to identify foreign antigens and produce antibodies against it. So that's what an antibody is. Antibodies are plasma proteins, as we've seen, GABA globulins, that are produced by the immune system in response to a foreign antigen. In an incompatible blood transfusion, there would be an incompatibility between what are the antigens that are found on the surface of the red blood cells and the antibodies found in the plasma, circulating in the plasma. So something to keep in mind here is that these antigens, when we're talking about blood antigens and blood antibodies, the antigens are found on the surface of the red blood cell, while the antibodies are circulating in plasma. The process of agglutination, that is the clumping of the red blood cells, where in an incompatible blood transfusion, so a blood transfusion that was wrong, okay, and that's when an antibody encounters the antigen and starts the clumping process that is known as agglutination. There are tons of antigens on your RBC membranes, okay, about 33 of them. But the ones that are going to evoke or that might lead to a serious transfusion reactions are your antigens A, B, and the Rh antigen, also known as antigen D. O is really not an antigen. O means that there are no antigens in there, so it's more like of a zero. So the antigens that evoke a serious transfusion are antigens A, B, and antigen D, also known as the Rh antigen. So let's take a look at the different blood groups and how are they named. So which one has which antigens and which antibodies. Again, the antigens are found on the surface of the red blood cell, and these are carbohydrates. We have either antigen A or antigen B, while the antibodies are in the bloodstream. These are plasma proteins circulating within the bloodstream. Within each person, you want to make sure that the antigen they have is totally different than the antibody they have. So if they have antigen A, their plasma should not have anti-A antibodies, because that means that that antibody is going to attack their red blood cells. And the blood groups are named according to the antigen, not the antibody. So on the surface of the red blood cell, if there is an antigen A, that means that that blood type is blood type A. In their serum, they would have anti-B antibodies. In blood type B, they would have B antigens and anti-A antibodies in the serum. Blood type patients or people with blood types AB would have both antigen A and antigen B, but they would not have any antibodies. So neither anti-A nor anti-B antibodies would be circulating in their plasma. Blood type O means that they have none of the antigens, so neither antigen A nor antigen B, but they would have both kinds of antibodies. They would have both anti-A and anti-B antibodies. And here you can see the different blood types again. So again, the name depends on the type of antigen found on the surface of the red blood cell. So you see here antigen A, that makes this type type A blood, and you can see the anti-B antibodies. Type B is the same, but if you move on to this type AB, you can find that it has both types of antigens, A and B, and no antibodies. Type O blood, O again stands for 0 antigens. So you see the surface of this red blood cell has no antigens on it, but it has the two different kinds of antibodies, both anti-A and anti-B. Now, agglutination again is when an incompatible blood transfusion happens, and now the antigen is in contact with its antibody. Okay, that is going to lead to the antibody attacking that antigen on the surface of the red blood cell, leading to clumping, a process properly known as agglutination, and you can see how that blood is going to clump or agglutinate, and that is a serious complication that could happen in incompatible blood transfusions. So what are the compatible blood types to give to patients? Well, obviously the preferred blood type would be the exact same type as the recipient. What if it's not available? Okay, so blood type O is pretty safe for everybody. So type O is considered the universal donor. So you can see here whether it's type A, if the recipient is AB, or O, they can all get blood type O as a second best option. So type O is your universal donor. O. Okay. Now for type AB, they are the universal recipient. They do not give blood to anybody. Okay, but they can take blood from everybody else. So it is safe for a person with a blood type AB. Obviously, again, your first choice would be AB, but if it's not found in the blood bank, types O and AB are safe options for these patients or for these people. Type O really does not have other alternatives. They can only get type O. This is a little video that kind of shows you the, goes over the different options. Okay, which one, the preferred again would be the exact type, while the other options or second options, what those are. And it's not just about the A and B antigens. You have to take into consideration the D antigen, which we are going to be talking about in a minute. So this is something that you guys will be doing in the virtual lab to figure out what kind of blood type you're looking at. So the test is, you put a drop of blood on a slide, okay? So we get a normal slide and you just put 2 drops of blood, one on the right and one on the left. And then you get this anti-serum. So this is anti-A, they're color-coded, and this is anti-B. And you mix them together and you look for the agglutination. If you find that the agglutination happens with anti-A, that means that this person is type A. If it happens with the anti-B, that means that person has type B blood. If it occurs in both of them, then that is a type AB. Well, if there's no agglutination that happens, that means that this patient or this person has a type O blood. This is a breakdown of the frequencies of the different blood types, broken down by ethnicity. These numbers, you do not obviously have to know them. It's just there to kind of show you that type O is the most common kind of blood, regardless of ethnicity, and type AB is the least common kind. Now for the Rh blood group, which is also known again as the antigen D. So when you get your blood typed, you are told, for example, you are blood type, say O positive or O negative. That positive and negative is really referring to this Rh antigen. It is named Rh because it was first discovered in rhesus monkeys, and then we found out that we have them too, so we still call them the Rh antigen, or again the antigen D. If somebody has that antigen, they are considered to be Rh positive, which means they do have the antigen D. If they do not have it, they are considered Rh negative, so they do not have the Rh negative antigen on the red blood cell membrane, but they also do not have the antibodies. Okay, and I put that kind of all in caps because it's not like the AB antigens, where if you don't have the antigen, you would have the antibody. This is very different. If a person is Rh negative, they don't have the antigen, and they also don't have the antibody. The antibody can be formed only if they are exposed to the Rh antigen for the very first time. The body will recognize this foreign antigen coming into the body and produce antibodies against it. Okay, so normally anybody that is Rh negative would not have the antibodies unless there was a prior exposure to that antigen, whether due to an incompatible blood transfusion or during a pregnancy, which we'll talk about in the next slide. And the seriousness of an Rh blood group, again, really is a serious thing to take into consideration because of a condition known as Erythroblastosis fetalis, or the hemolytic disease of the newborn. So one of the routine exams to be done when a woman is found to be pregnant is her blood type. And we're really looking for that antigen. Okay, Rh positive, an Rh positive mom is a safe mom in terms of blood typing. What worries us would be an Rh negative mom pregnant with an Rh positive fetus. So know that the gene that codes for the Rh protein is a dominant factor, meaning that if mom is negative and dad is positive, the baby is going to be positive. So it's a dominant trait. So again, when you want to worry is if the mom is Rh negative and the baby is Rh positive. An Rh negative mom means that she does not have the Rh antigen and she has never been exposed to it. The baby, though, does have that antigen on the surface of its RBCs. That first pregnancy is going to be totally safe. No mixing of blood occurs during pregnancy. It only occurs during labor. So during childbirth, there is a little bit of mixing of the blood between mom and baby. And that is when mom is going to be exposed to the very first time to that new antigen, which is foreign to her, and it will stimulate her immune system to make antibodies. But again, this is during delivery, so that baby is going to be safe. Nothing is going to happen to that baby. You can see her now after delivery with the antibodies found in her bloodstream. The danger would be in the next pregnancy. So again, if that second baby is also Rh positive, now these antibodies are going to cross the placenta and attack the red blood cells of that second baby. And that could be fatal, hence the name Erythroblastosis fetalis, because it happens in the fetus, and where it'll blast these red blood cells, literally destroy them. And again, it can lead to the death of that baby. So erythroblastosis fetalis is a very serious condition, and usually moms are given, if they are Rh negative, they are given what is known as a RhoGAM injection that kind of protects them and blocks their immune system from making these antibodies. And this concludes chapter 14.